Hey, welcome to the Gladden Longevity Podcast. This is Dr. Gladden here. I just want to let you know we just finished a conversation with a really interesting guy, Christian Drapeau, who's been in stem cell research for the past 20 years. And we just had a conversation that was very, very far-reaching on different kinds of stem cells, the best environment in which stem cells need to survive and thrive, stem cell exhaustion, stem cell depletion. We cover many, many topics that you're going to find incredibly interesting. And out of this, actually, we pull together a paradigm of how stem cells are best used. And we both ended up agreeing on this paradigm. So I think you're going to find this a really, really interesting conversation. Welcome, everybody, to the Gladden Longevity Podcast. I'm your host, Dr. Jeffrey Gladden, and I'm here by myself today. Steve is off playing hooky someplace. He didn't tell me where, but I'm sure it's not work-related, although he said it was work-related. That being said, we have a great show today. I'm with a gentleman who I've met a couple times now, Christian Drapeau, and he's a really, really interesting character with some interesting information about stem cells, which is a hot topic, of course, in regenerative medicine, longevity medicine. So with that, Christian, welcome to the show.
My pleasure. Thank you.
Yeah. So tell the audience a little bit about your background, just to sort of set the stage, if you don't mind. I know you have a master's and you have some other things. Just kind of walk us through a little bit of your background to give them a sense of who you are.
So my background is in neurophysiology, so brain research. I was working at the Montreal Neurological Institute. I was working on my PhD, essentially on epilepsy. And events happened in my life, so I quickly wrapped an almost completed PhD into a master. But I came back to the whole world of research a few years later when I started to was hired by a company selling blue-green algae from Klamath Lake and to study the effect of that product in the body. So that's what brought me back to the whole world of science.
What did you find with the blue-green algae? What was it doing?
Well, this was right after the passage of DSHEA, the Dietary Supplement Health and Education Act, and we had companies selling a product needed to have scientific data to support the claims that they were making. So really, I was hired to document the mechanism of action for that plant or blue-green algae. And we very quickly identified the active compounds for its anti-inflammatory effect, for its effect on the immune system, and for its effect on the brain, concentration, mood elevation. And as I'm doing all of this, I came across people reporting benefits with multiple sclerosis, liver failure, diabetes, emphysema, heart disease, Parkinson, liver failure, kidney failure, joint problem, scars that would disappear. So the big question from a scientific standpoint was, what is this thing doing to help someone's heart, someone else's pancreas, someone else's lung, brain, and so on? So we did a number of projects. We got good data, studies. We got good data, but nothing that really ever gave us like the real aha until I came across an article documenting that stem cell had the ability of going to the brain. The title was Turning Blood into Brain. You remember early 2000s, stem cells are only blood precursors. They don't become other tissues. And of all things, the brain is not the one that you would think, regenerates out of typical traditional brain science. So the fact that a stem cells was seen to go in the brain and becoming a brain cell, I thought was quite groundbreaking to me. So pre-internet, so I went to the local library, looked into microfilms, and looked at other articles that may document a similar phenomenon, meaning stem cells going into a tissue and becoming cells of that tissue. And I found one in the liver, one talking about the heart. So I thought, if stem cells can become heart, liver, and brain, why not pancreas, lung, skin, and the rest? Makes no sense that they would become those three and not the rest. And if they do become all the tissues, which was the hypothesis, then they have to be the repair system of the body. So we published...
Was the blue-green algae's effects, was it in some way stimulating stem cell activity? Or
That's what it was. So we make this hypothesis that what if that plant was actually stimulating stem cell release? That was sort of the whole background of this thought process. Nobody had ever looked at this before because stem cells were not a thing. So we bought a flow cytometer, started to count our own stem cells, and we discovered that's what this blue-green algae was doing.
Okay, that's interesting. So just so the audience understands, there are different sources of stem cells, right? And we might just sort of clear that up for the audience. So what we're talking about here are hematopoietic stem cells that come out of bone marrow, stem cells that are traditionally slated for making, blood products, red blood cells, et cetera, right? Clotting factors, or platelets. And then you've got mesenchymal stem cells, which basically are typically found in fat tissue, which also are capable of aiding repair. There's always a question whether or not these stem cells actually become the actual tissue or whether they actually stimulate the local tissue's progenitor cells, which are kind of like the local tissue cells, to actually do what they do better. It'd be interesting to know if you had like a DNA fingerprint where you could say, well, this was in the bone marrow, now it's in a neuron, versus it was in the bone marrow, now there's more neurons, and maybe it was the brain, progenitor cells or something else that came together to actually create that neuron. But just so the audience understands, there are different kinds of stem cells, and you have these progenitor cells really throughout the body in lots of different tissues. Like your blood vessels have progenitor cells, your heart has progenitor cells, your lungs. And these are cells that kind of sit in reserve that can actually repair that local tissue. Then you've got mesenchymal stem cells that can interface with multiple tissues. Hematopoietic stem cells can interface with multiple tissues. Then you've got embryonic stem cells, which can basically start to interact with all tissues and can become more pluripotent, right? And then problem with some of those is that if you get too embryonic, too differentiated, they can actually be associated with tumor formation.
Correct.
And so there's a whole spectrum of stem cells in terms of, they can do everything to they're more focused on rebuilding this particular tissue. And the trick, as you're thinking about stem cells, is to get stem cells that have enough general utility that they can be used, but not so de-differentiated that they can turn into tumors. And that's really kind of how to think about this, right? So,
correct.
Yeah. So go ahead.
I was going to say, and that's really the big discovery with adult stem cells. They're not as potent as embryonic stem cells. They're not as powerful, but they're not linked with tumor formation like embryonic stem cells, have been linked to. Right. The stem cells that we're talking about here that are released by plant extract that we have documented over the years, they are mesenchymal stem cells. They're both endothelial progenitor cells, mesenchymal stem cells, V cells, some hematopoietic stem cells as well. Okay. Just a small parenthesis on what you mentioned before. Mesenchymal stem cells originally come from the mesenchyme, which is essentially the bone marrow. Okay. So they are in the bone marrow. And what is very interesting is that the way that they were isolated originally is that if you take a sample of the bone marrow, put that in a test tube, and you just swirl the test tube, if you rinse the test tube after, the stem cells, the mesenchymal stem cells, stick to the glass. So it's a very easy way of isolating them. And then later on, they realize the stem cells in fat tissues also stick to the glass. So they were called mesenchymal stem cells, but they're not from the mesenchyme. So in a way, the term mesenchymal stem cells now being attached to many, many different types of stem cells in the body. They are multipotent, pluripotent, however you want to look at it. And as you said, stem cells, oftentimes get into a tissue and stimulate the tissue resident stem cells. But there are many, many studies that have shown that stem cells can also convert into cells of those tissue. And that is done by tagging stem cells with something like green fluorescent protein. So that stem cell has the gene, it starts to make green fluorescent protein. And when that stem cells become cells of a muscle, for example, then the muscle is all fluorescent. Right. So we did a study like this. We published this in 2010, where we injected cardiotoxin in the tibialis muscle of a mouse, which basically killed the muscle, and then gave this blue-green algae extract to the mice. And then we had a control group beside to look at the enhanced repair by releasing stem cells. And what we saw is that after 6 weeks, the entire muscle is reconstituted, but it's completely green. Meaning it is entirely emerging from bone marrow stem cells.
Interesting.
And now you go in the brain, and in the brain, in the heart to an extent as well, you will see very few stem cells, very few new cardiomyocytes that are green, meaning they're not derived from stem cells. But if you feed these animals like BRDU, for example, that will make you tag new cells, you realize that there are a lot of new cells in the heart, but they have been stimulated by stem cells, but not emerging from stem cells.
Right, exactly.
So you got the whole spectrum here of how the body repairs.
Yeah, exactly. So I was reading an article recently. Actually, I came across it not even thinking about the fact we were going to have this conversation, and I saw we were about to have this conversation, I thought, oh, that's interesting synchronicity here. But I was reading about an article how a decline in IGF-1 signaling. And just so the audience understands, IGF-1 is basically something that is kind of a anabolic messenger, if you will, in the body. IGF-1 is really growth hormone, gets processed in the liver into insulin growth factor 1. And insulin growth factor 1 then goes out and does the work of growth hormone to stimulate growth, to stimulate cell division, repair, things like that. It can also stimulate cancer. And so what was interesting to me was that they showed in mice that as mice became middle-aged, their hematopoietic, their bone marrow stem cells, started to decline in number and potency, and it was directly related to a drop in IGF-1. And when they actually replenished IGF-1 in these middle-aged mice, rather, they could reboot the activity of those hematopoietic stem cells as bone marrow stem cells, which I thought was really pretty interesting. I hadn't seen that previously. Is that something that you were aware of, or?
Yeah. There's a number of these growth factors. It's not
That's right.
just IGF. There's a number of these growth factors that have been associated to the same kind of decline.
Yeah.
Whether it is causal, I'm looking at it from let's take a lot of distance from the whole situation and look at it. Let's look at it very general.
Mm-hmm.
We have a decline in the number of stem cells because the red marrow shrinks into yellow marrow or fatty marrow that does not make stem cell. That process is natural. It happens in everybody. By age 30, we've lost about 90% of our red marrow, which coincide with this realization in our 30s that, you know what, we're not Superman anymore. Like we don't repair like we used to. We don't have as many stem cells available to repair. And that is followed also by a decline in a lot of these different growth factors. And somewhere, and this is purely hypotheses because I don't know if there's ever a way to know, but the point is that if we were to say we have stem cells that keep multiplying like when we were young, and they keep going like this all our lives, as cells start to get damaged as we age, then suddenly the duplication process could probably increase dramatically the risk for tumor formation. So somewhere it's almost like the body, as we age, reduces the ability, the boost for that cellular division to avoid risking to have tumor formation. So I'm not sure, and I'm saying this just to say the big question is, should we then replenish things like IGF in the body to boost stem cell production? Aren't we going to just create another problem that is even worse?
Yeah, well, that's a really interesting question, right? So you may be familiar with the TRIM trial, where they basically were looking at middle-aged men and they gave them growth hormone, DHEA, and metformin. And over a year's period of time, they reduced their biological age by 1.5 years, which at that time was really pretty spectacular. But now we can drop DNA methylation ages by 10 years in a week, basically, if you do a 5-day fast mimicking diet. So, but anyway, the point was that they also did MRI scans of the thymus gland, which also involutes, and along the lines of the bone marrow declining, right? The thymus gland involutes, which is responsible for putting out thymus cells, T cells. And with the growth hormone that they were administering, they showed that the thymus, which had been infiltrated with fatty tissue, was actually regenerating. They were actually regenerating the thymus gland. And so the question was, is growth hormone the fountain of youth because we're regenerating the immune system? And now we're talking about maybe growth hormone / IGF-1 rebooting the bone marrow, rebooting,
it does,
right? Which it does. And then rebooting these stem cells, right? So of course, what we're alluding to here is there's a flip side to growth hormone and IGF-1, which is if you're exposed to it all the time, you do have a higher risk of cancer and people don't live as long. And that's well documented also, right? And there's good reason for that. We won't get into all the biology of that right this second. But to my way of thinking, particularly when someone is injured, one of the things that we like to do is to boost stem cell release, boost stem cell function, and then maybe even put in some of the peptides that will actually stimulate growth hormone release, do some things to boost IGF-1, some things like that to kind of scooch the whole system along during the healing process, but then not be doing that continually, right? Maybe pulse that on and off periodically and then combine it with things like rapamycin, where you're really inhibiting the effects of IGF-1 and mTOR, right? Mm-hmm. So I think biology, being this economy of balance, likes to cycle in and out of states. It doesn't like to be in one state all the time. And I think what we're working with right now is the concept that this becomes the healthiest way to go forward, because without testosterone, growth hormone, IGF-1, stem cells decline, thymus declines, lots of bone density, muscle mass, all that decline. And yet we know that if we're on it all the time, it's a bad thing. You want to activate AMPK, you want autophagy, you want telomere, repair, you want cellular repair, mitophagy, all these things that rejuvenate you, that are independent and actually only occur in the absence of growth hormone and IGF-1, right? So you need both sides of the teeter-totter, so to speak. Mm-hmm. For us, it's really become this question of how do you best cycle people between that, and where are they in the aging process? What is their cancer risk, and how do you do that in the most wise fashion, if you will?
Yeah. The way that I'm looking at stem cells is because it was studied as the repair system, like the study we just mentioned before that we did when we triggered an injury to a muscle, monitor and observe the recovery of the muscle, document how much that repair is actually driven by stem cells. So all the studies were about studying the repair process of stem cells. But as we did these studies, like for example, that study that we did on the muscle, we looked at the muscle, but when we started to look at all the other tissues, we also find green cells in other tissues. They did not all go just to the place where there was an injury. Nice. And if there is no injury, like we did not do this, but other groups did this. So you have these mice in that, let's say the same study that we did, and you're left with 3 mice at the end that you don't need to sacrifice for the study. They're extra, and you keep them as pet in the lab. Yeah. And 6 months later, you go and you look at them, and now you realize that they are green cells, like entire organs, completely green. And what is emerging from this, same thing was done in humans. You look at women who get a treatment for leukemia, so they get irradiation and then injection of stem cells from a male compatible donor. So that means now their bone marrow is all made of stem cells with the Y chromosome. Mm-hmm. So if this phenomenon happens in human, let's go and see biopsy tissues that were taken at the autopsy of a woman who passed away after that treatment, 5, 10, 15, 20 years after that treatment. Yeah. And now looking at the time since bone marrow transplant and across tissues, the whole picture that is emerging is that we have a new liver every 2-3 years, a new skin every month, new lining of the intestine every 5 days, half of a new heart every 25 years. So everything is in turnover. Yeah. So turnover means I lose cells every day. So to stay healthy, I need to replace these cells. But what happens now if you've lost 90% of your stem cells by age 30? Yeah. There's a point when we pass 30, we just don't have enough stem cells to replace the cells that are being lost. And there's this cumulative deficit in tissues and organs. So so the idea, and that's really the sort of the past 20 years of research that I have done, let's not leverage growth factors to stimulate stem cells. Let's just stimulate their release from the bone marrow as if we were mimicking a mild injury. So it's almost like an injury mimicking process.
This episode of the Gladden Longevity Podcast is brought to you by Mimeo, a supplement designed to give people the benefits of fasting without having to fast.
Yeah, Mimeo's research found that the human body produces a unique set of molecules during a 36-hour fast. And those molecules were responsible for activating many of the cellular health and longevity benefits of fasting.
With Mimeo, you can get the cellular health and metabolic benefits of a 36-hour fast even during a meal, or you can use it to maximize the benefits of your shorter fasts with powerful boosts in mood, focus, energy, performance, recovery, and appetite control. Dr. Gladden, in the 2 years I've been working with you and the 5+ years I've been listening to the show, I've never heard you talk about something outside the Gladden Longevity line of products like I've heard you talk about Mimeo. Why do you like it so much? Use it and make it a part of some of your presentations.
Yeah, we love it, quite honestly. I mean, the idea of Mimeo being able to mimic fasting, we all know the benefits of fasting, is dramatic in and of itself. And even in the context of if you're eating, and if you're fasting, it even augments your fast that much more. But the interesting thing is that it also decreases things like neuroinflammation, it suppresses appetite, and it increases a very, very important biological process called reverse cholesterol transport. And that's critical for cardiovascular disease, for decreasing risk of dementia, cancer, et cetera. It's really, it's quite a fascinating product. I've really been kind of blown away by it.
As a way to help support this podcast and save money on your Mimeo order, you can use the discount code GLADDEN10 to get 10% off your order at mimeohealth.com.
Think of it as a way to unlock the power of your biology. Let me ask you this. So if the strategy that you're talking about here is really devoted to releasing stem cells from the bone marrow, which certainly can be advantageous, then it raises two questions. One, if we're not doing anything to keep the bone marrow youthful with a large number of these stem cells, are we just depleting them? And then the other thing becomes, if we're basically using things to basically release these stem cells, are we just kind of using up our supply that much faster? So,
I mean, it's a totally legitimate question, because that's the first thing that came to mind
Right.
in 2001 when this whole concept came up.
Right.
And there are many ways to answer the question, but the answer is no. And I can approach it from various angles. Number one, to understand that while the red marrow shrinks into fatty marrow, so stem cells, marrow that does not make stem cells, the marrow that you have, the red marrow that you have, produces stem cells continuously. When what is happening in the bone marrow, contrary to everywhere else in nature, where you have symmetrical cellular division, meaning one mother cell becoming two identical daughter cells, in the bone marrow, you get asymmetrical cellular division. So the original DNA goes into one daughter cell that stays in the bone marrow, and the two copies go into the other cells that will be released, so that you maintain your original DNA. Because if the bone marrow didn't do this, after 5 years old, you would have so much copies of a copy of a copy of a copy. We would be like a ambulant mass of tumors.
Mm-hmm.
So it protects the bone marrow. So the consequence of this is that when you release a stem cell, it left a sister behind. So when we release 10 million, I don't think you have 10 million fewer stem cells in circulation.
Okay. What you're saying is you're stimulating the bone marrow stem cells to divide and then release. You're not just releasing them, so to speak.
I think we release them, but the release immediately is accompanied by a division. Okay. Our effect, the active compound, is having an effect on the release.
Okay.
But I think duplication is associated with it. Right. The other way to answer it, two other ways. Look at who are the people in our society that release a lot of stem cells. Athletes. Mm-hmm. You train really hard, you will significantly increase the number of stem cells in circulation. Right. Are athletes walking in their life one day and just running out of stem cells? No, it's not a phenomenon. You multiply them. Yeah. So it's not a reality. And lastly, all those ingredients that we have identified have an effect on stem cells. They actually turn out to be ingredients associated in folks' medicine historically with longevity. They've not been associated, any one of them, with some sort of side effect of exhaustion of something of some kind. So nowhere in the scientific literature, like I'm not aware of one piece of data that would point to the fact that there could be such a phenomenon as taking too many stem cells.
One of the hallmarks of aging that was originally talked about back in 2019 when the first 9 were published, I think we're up to about 16 now, was not only cellular senescence, but stem cell exhaustion. Mm-hmm. Right? This concept of stem cell exhaustion, where we do run out of viable stem cells, because stem cells themselves can age over time, secondary to their exposure to, let's call it, environmental factors, whether it's radiation, chemicals, whatever else, plus the aging process. And so the question I would have is, do you think that this molecule that you're using has any impact, or have you studied whether it has any impact on stem cell exhaustion?
It's tied a little bit to what we just talked about. I think that the word stem cell exhaustion has created quite a bit of confusion, in the sense that I think we're tripping in words, in the sense that we use the same word for two completely different mechanisms.
Yeah.
One is stem cell exhaustion in the sense of stem cell depletion in tissues, just like we talked about before earlier on when you were describing how stem cells get into tissue and then through a paracrine effect starts to stimulate the tissue-resident stem cells.
Right.
If a tissue is subjected to a chronic stress and a chronic wound that it is trying to repair, that tissue will run out of its resident stem cells. That tissue will reach stem cell exhaustion. That is the stem cell exhaustion that leads to an actual problem in that organ. You don't reach stem cell exhaustion in the bone marrow by using your stem cells. You have stem cell conversion, where red marrow converts into yellow marrow, but that would be calling natural human evolution a problem. It happens in every single individual. I will even go as far as to say, if we do reconversion, like preventing or reversing yellow marrow to red marrow, that will be your first red flags for leukemia. So right now, this conversion is a normal process, and it does not exhaust in the sense that we can die saying that our power to repair has gone down, but nobody dies not having any stem cells left. They continue to play a role of repair during their entire life. We have people with heart disease taking our plants that trigger stem cell release in their 70s, 74, 78 years old, and they reverse their heart disease just by releasing their own bone marrow stem cells. So at that time, the system is still effective in their body. So I think stem cell exhaustion, we need to understand, is really stem cell depletion in the tissue. And the solution for this is to release more of our bone marrow stem cells that will go and replenish these tissues.
Do you want to comment at all on very small embryonic-like stem cells, VSCLs?
Yeah, they're fascinating. They've been discovered, just to give the whole story, they've been discovered by several groups. One group called them blastomere-like stem cells. Blastomere is the early embryo. So they are in the body, they look like, and they have about the same regenerative potential as embryonic stem cells, but they don't lead to tumor formation. And then another group called them very small embryonic-like stem cells. Essentially what they are is that they are stem cells that have been historically discarded because they're too small. They're the size of a platelet, essentially. So you take the platelet-rich fraction or plasma, it's filled with V cells that were considered before to be simply platelets. But if you tag them for DNA or tag them for stem cell markers, they are full cells with their DNA and with stem cell markers. The beauty about V cells is that they are a type of stem cells that can be in a dormant state or in an activated state. They have been documented by some researchers to be released from muscle mass, muscle tissue when there's an injury. They're also found in the bone marrow. Their main claim to fame is that they are really the most potent stem cells that we have in the human body.
My thought was that in understanding them was that the V cells actually are the stem cells of the stem cells, so to speak. So that when you do, let's say your bone marrow does start to become, the very small embryonic-like stem cells become the reserve of the reserve to help rebuild progenitor cells and bone marrow stem cells, et cetera, so that it's kind of like your secret reserve. And, Todd Ovakaitis basically developed a laser to kind of liberate these VSCLs in that PRP-rich fraction of the blood from a protein that was binding them and holding them in suspended animation, if you will, which is interesting. There are other technologies and techniques that are used to do this too. I know cold exposure has been used to liberate or activate very small embryonic-like stem cells. But the point is that when you're using the product, this blue-green algae, you're releasing the stem cells from the bone marrow, but not really having a direct impact on the release or activation of very small embryonic-like stem cells. Is that correct?
It is correct, but we did some work earlier on, probably 2007 to 2010, with this first blue-green algae extract. And what we were able to document, and actually we see the same thing with a lot of the plants that we use that trigger stem cell mobilization. Right now we monitor mesenchymal stem cells, endothelial progenitor cells, premature versus more evolved stem cells. And we also monitor stem cells by size. So V cells, or we call them small cells in how we quantify them. So these plants also increase the number of small cells or V cells in the blood. And in some preliminary work that we did, it was never published, but we also see that the number or the proportion of them that are in their activated state has significantly increased. Like about 30% of them have switched to their activated state. We have stopped going in that direction. Circumstances, we should go back to it.
Mm-hmm. Yeah. Yeah, it's interesting because if when we're mobilizing stem cells, we're actually mobilizing more than stem cells. One of the things that we think about when we're utilizing STEM products is the environment into which they're being placed. So there's data that shows that if you put, healthy stem cells into old plasma, they don't divide, they don't do things as well as they could as if they're in younger plasma. So kind of cleaning up the environment, things like plasmapheresis, young plasma, things like that to kind of prep the landscape, and then you put in, the Olympic swimmers into the clean swimming pool, and now let them go to work, right? And so, depending on who we're working with, this is a strategy that we use also to optimize stem cell results, and then we combine it also with signaling peptides and other things that support what they're doing, right? So you create a whole ethos for them.
Yeah. What we have seen in an opportunity that we had, probably this was maybe 2002, 2003, and we had access to a number of Alzheimer's patients. And the idea that we had at the time is, let's count the number of stem cells in their bloodstream. And we were expecting to see fewer stem cells in people with more advanced Alzheimer's. And that's not what we saw. To our surprise, we did not see fewer stem cells. What we saw, however, is that the more advanced Alzheimer's patients, their stem cells had a different phenotype. The adhesion molecules needed for stem cells to migrate into tissue, those that are triggered by the signaling coming from a tissue when the stem cells goes into the fine capillaries of a damaged tissue, those signaling molecules and adhesion molecules were lost. They were no longer on the surface membrane of stem cells. However, the advancement of Parkinson was associated with systemic inflammation. So the idea was essentially that your inflammatory signal that triggers the migration of stem cells is no longer happening in the postcapillary venule of an affected tissue, but it has become systemic. So when the cell reaches the capillary, it has spent its mechanism of migration. So now these cells can no longer reach the target tissue. So along the line of what you were talking about, to me, before a treatment, a stem cell treatment of any kind, I would suppress systemic inflammation for a few weeks before the injection. And we also need to understand that these stem cells can be fairly large compared to a capillary. So we also need to boost capillary size, nitric oxide. We need to have better fluidity of blood, like nattokinase. We need to have better sliding, if you want, passage of stem cells into capillaries. So you rebuild the glycocalyx, those kinds of events to be able to support all of that prior to a stem cell treatment. So we're developing products right now that will do exactly that. So when you take STEMREGEN, release your own stem cells, you also support their role in the body.
Yeah, I think it's a critical point that you make. A stem cell is just a tool. It's a bullet, but you need a whole army around it to make it effective, right? And
correct.
So we do a lot of that with glycocalyx optimization, with nitric oxide production optimization, things like that, to improve not only the cardiovascular system, but blood flow to these areas. And then, plasmapheresis is a wonderful way to decrease inflammation and remove inflammatory cytokines, right? So that becomes a really nice prep for, what we're talking about. And depending on how sick somebody is, it may take 2, 3, 4, 5 sessions of plasmapheresis to kind of get them down to a point where now the stem cells can actually really be effective, right? So as you're listening to this as an audience, just understand it's not just about, there's a clinic up the street that does stem cells, I'll go in and get some injected, everything's going to be fine. It's really, you really have to look at this in a more comprehensive way, or I'm just going to take a supplement and everything's going to be fine. You've got to look at it in a comprehensive way. So, yeah.
Correct. Yeah. I mean, the studies that we have done with the plants, because we are not a clinic, we're not there to act medically, if you want, in any way. So the simple fact of taking a supplement that will put more stem cells in circulation, what is the impact of that if you do nothing else? Understanding that if you do other things, the results will be better, faster, everything will be better. But what do we get if we just put more stem cells in circulation, understanding, as we talked before, that more stem cells means a greater ability to repair? So what can we reach as outcome? And we have an ongoing study right now that is done in Madrid and Miami with the cardiologist. And we chose this because historically, we have seen a lot of benefits with the heart. And we are now ongoing with this study. It's still moving on, but we have 10 patients that have gone through the whole cycle. And of those 10 patients, all of them end up after 6 months with normal cardiac function. They could certainly get better.
What kind of heart disease are we talking about? Is it
congestive heart failure? So they start with stable chronic congestive heart failure, at least 2 years, controlled with everything that you do for congestive heart failure patients, beta blockers, diuretics,
and
all of that.
Are these hearts that are enlarged? I mean, there's many kinds of congestive heart failure, right? So is it
right now,
congestive heart failure or?
The only thing that we followed, call it a preliminary trial. So we didn't go into the detail. They're all people that had ejection fraction lower than 43%.
Okay, that's low ejection fraction congestive heart failure. There's normal ejection fraction congestive heart failure too.
Correct.
Based on if you're listening to this for the audience, think of it this way: the heart has really two functions. One is it has to relax to receive blood coming in. And then it has to pump. It has to be strong enough to pump that blood out, right? And so you can have heart failure based on either an inability to relax or an inability to pump or both. So that's why you could have a normal percentage of blood pumped out, but if you can't relax to get it in, you're still going to go into congestive heart failure. So
correct.
If you have a reduced ejection fraction, then you probably have more of a mixed bag here where there's always some what's called diastolic dysfunction, decrease in the ability to relax, but also the primary problem at that point is the inability to pump. So
correct. We basically do ejection fraction. We do obviously all the questionnaires associated with heart disease. And the clinic has an interesting characteristic is that it is sitting on a hill. So they basically have the patient go down the hill and see how far they can climb on the hill before they need to stop. And they all started with some level of not being able to climb the entire hill. It's not a lot. It's maybe, 8 degrees maybe. And 6 months later, it's no longer a limitation.
Nice.
They can climb the hill and they're totally fine. They're not an athlete. They're just normal. The age bracket was 68 to 76 years old, I think. And they're just normal 76-year-olds.
We should do a trial like that. Our offices, as you know, you've been to them, are on a hill. So
there you go.
Plus, we do a little bit more sophisticated. We do, cardiopulmonary exercise tests where we can measure VO2 max and
let's do it.
Stroke volume to get us an idea of what's happening. Yeah, be fun to do that. Well, very cool. So, you alluded to the fact that you have this product that you came across, and it does have the ability to release stem cells from the bone marrow. And then you alluded to the fact that you're working on some other things. What are the other things you're working on? Some things related to blood vessel flow, glycocaine?
Yeah. So I've worked with this before in the past, and we saw indeed much better results. So you release stem cells, but they need to be able to circulate. We need to understand, for a stem cell to be able to do its regenerative role in the body, it can only leave the blood circulation into one very small area of the entire blood vasculature, and it is the post-capillary venule. It's the place in the blood circulation right when the capillary becomes a vein. There's a difference of pressure between a capillary and a venule, and that difference of pressure creates turbulence. Turbulence, through its mechanical force, activates molecule at the surface of stem cells that makes the expression of the receptor that listens to the signal that the tissue is sending if it is injured. If that signal is there, it triggers the migration process. That's the area where they can migrate. So if stem cells cannot fully circulate into fine capillaries because they're big, they're bigger than the capillary. They can go up to 20 microns. Your capillary is up to 12. So you need to have slick blood. If your blood is too thick, you do not facilitate stem cells reaching the fine blood circulation. So nattokinase. So we've played with this in the past, and we did see a number of cases that worked much better. We have this notorious case in the Philippines. He was the former president of the Breast Cancer Society in the Philippines, and he had a stroke, and he was paralyzed with aphasia. And the moment that we coupled the product that we had at the time with a product, simple nattokinase.
Yeah.
And within about 2-3 months, the whole thing was resolved.
That's
So
really
the
interesting.
product we're working on right now, we're trying to go beyond what we did before. So we're basically blending different aspects that are going to promote circulation. So nattokinase type enzymes to make the blood more fluid. Then we need to support the synthesis of collagen and the health of collagen around capillaries, because a capillary is not an actual tube that you can dissect. It's more like a hole in your collagen that is covered with endothelial cells. So the integrity of a capillary depends on the structure of the outside environment. Then we also have nitric oxide technology to help to relax the muscle cells and open these capillaries. And then glycocalyx technology to make these capillaries where cells can really sort of squeeze into them and slide.
Right.
So it's a product that brings these 4 types of approaches into one product.
Nice.
And the other one is, like we talked about before, systemic inflammation. So plant extract that are going to reduce like inhibition of COX-2 activity, lipoxygenase, inhibition of cytokine secretion, digestion of some of these enzymes. I mean, 4 or 5 different routes of inflammation that are suppressed with different plant extracts.
Right. No, it's interesting. Just so the audience understands. So this nattokinase is kind of, think of it as kind of dissolving. One way to think of it is to think of it as kind of dissolving clotting elements in the blood that would thicken it up so that you keep the blood kind of loose so that it flows. This glycocalyx that we're referring to, imagine a stream flowing, and at the bottom of the stream, there's like grass, right? And the grass in the artery, which is this proteoglycan glycoprotein that kind of lines the skin, the internal skin of the blood vessel, it actually has an electrical charge. So it pushes things away from the wall of the artery and keeps them moving through the lumen or the opening of the artery. So it makes for, now if you have blood that's sort of thinned a little bit with nattokinase and you have this slick tube, all of a sudden, even though the cell is larger than the opening, the cell can get through because it can deform in and stretch out and go through, right? So, but if you don't have these things working for you, it can get stuck there. And then it never really gets to where it's going to go, which is part of the problem that people talk about when stem cells are given IV. Don't they all end up in the lungs, right? Because they're going to go through the lungs first,
correct,
through the venous system and get theoretically stuck there. But if you're using these kinds of approaches, you can, yes, some of them will, but you can help them navigate through, right?
Yeah. I'm not sure if I've read all that literature about stem cells getting stuck in the lung. I'm not sure if I fully agree with it in the sense that, like you just said, a stem cell, its diameter when it is unchallenged is about 20 micron. But if you've got like good consumption of omega-3s with a very flexible cell membrane, they squeeze, that's their function. They squeeze and they will go through it. And the way that I oftentimes explain glycocalyx, like you did, is when you find an old pipe in a field at the end of the summer, you just put your finger inside and it's like slick, like there's a algae that has grown on it. It's like very slippery. That's your glycocalyx. It makes anything that could get stuck in that pipe just slide in that pipe because it's slick. And that's what the glycocalyx does. And it's essential for proper functioning. Not only it's essential, I will go beyond this, but the research is showing that the start of what becomes atherosclerosis and all the arterial disease start by a breakdown in the glycocalyx 100%. That leads to a small injury. And if you don't have enough stem cells in circulation to repair that injury, then the cholesterol and all the rest of the stories become something that you need to deal with. But if you have good glycocalyx and good number of stem cells in circulation, the problem is never there.
That's right. And just so the audience understands, we destroy the glycocalyx when we eat sugar. High sugar destroys the glycocalyx. So it destroys it like right now. I mean, in a matter of minutes. And we actually did a trial in our office with a product, an algae product called Arterosil, and we showed that you could restore the glycocalyx in a matter of minutes. Actually, it can be destroyed and it can regrow quickly. Like we tested at baseline 30 minutes, 60 minutes, 90 minutes, 180, and I think maybe 240. And virtually 85% of everybody had dramatic improvement in arterial elasticity, suggesting the glycocalyx was back working normally or restored. So just understand that this is very dynamic. And so in my own world, when I'm eating, I take Arterosil because that's when I'm going to be destroying it. It's when I'm eating, right? It's not when I'm running. So anyway, interesting stuff.
Yeah. So all of this, you blend all of this, and I think then you maximize the role of your stem cells in your body. Because at the end of the day, we can talk about all the science, you can go into the details. To me, what is emerging from everything in stem cell research is that repair means, like, when you have a cold, you need your immune system. Generally speaking, you just need your immune system. When something is broken, you need the building blocks of those tissues. These are your stem cells. Anytime there is repair that is taking place in the body, it always involves stem cells. Before, we didn't know about it. So you use red lights, pulse electromagnetic frequencies, tons of things that we do that stimulates repair. But at the bottom, it just utilizes stem cells better. That's why these technologies are working. So the building blocks, the basics of repair is stem cells.
Yeah. No, that's right. It is interesting you mentioned red light and PEMF. I was just exposed recently to pulse magnetic induction, a pulse magnetic induction mat. And I've used PEMF many times. I had something going on with my lower back from basically doing a rowing exercise where I kind of strained it a little bit. And it kind of been nagging. Anyway, I was on this thing for literally 20 minutes and it restored all the strength in my back. Like, I got up the next morning, I could do an exercise I hadn't been able to do for some number of weeks. And I had like full strength back. I was like, wow, that's amazing. So to the point here, if you're combining, the quote unquote stem cells or stem cell release with these other technologies, this is how you really get kind of fantastic results. So that's
correct. Yeah, the technology you just talked about has been like very well documented to boost capillary flow. So the moment you boost capillary flow, it's circulation of not only stem cells for repair, but it's oxygen, it's nutrients. I mean, it's everything. Everything in your body is circulation. More circulation means more seepage of plasma into your tissue. That means greater lymphatic return. That means greater cleansing of your tissues. So everything works better.
Yeah, beautiful. Well, Christian, it's been a fascinating conversation. Really appreciate you being with us. I think it kind of expanded beyond what I thought it might. So I always like a good conversation like that. So thank you.
It was very nice. Thank you.
Thank you for listening to this week's episode of the Gladden Longevity Podcast. If you would like more information on what we've discussed or other topics, please reference the show notes or go to GladdenLongevityPodcast.com. You can also find us on Instagram, Facebook, and Twitter by searching Gladden Longevity. If you've enjoyed this podcast, please subscribe to get future episodes delivered to you and share our podcast or this episode with someone in your life that may find benefit. Thank you for listening. We'll be back next week with another exciting episode. The Gladden Longevity Podcast is provided for informational purposes only. It does not constitute medical advice. This content is not intended to be a substitute for professional medical advice, diagnosis, or treatment. Always seek the advice of a physician or other qualified health provider with any questions you may have regarding a medical condition. The use of any information and materials linked to this podcast is at the listener's own risk.